On 4th of January 2021, the Department of Health and Social Care announced that the first few people received the Oxford University AstraZeneca coronavirus vaccine, which marked the start of the NHS COVID-19 vaccination programs that are rapidly expanding across the UK. So today, I will be explaining the science behind this viral vector vaccine and how it works. I will also look into the effectiveness of this vaccine, briefly looking at clinical trial results, and finally, is there any clear side effects and safety issues with this vaccine? Please, right now, before we dive into this video, like, subscribe and share this critical information with others. If you have any questions regarding the science in this video, your thoughts on this vaccination program, as well as suggestions for future videos, drop it into the comment section below. Beginning with the science behind this viral vector vaccine, the flow of our genetic information moves from DNA to mRNA to protein within our cells. The SARS-CoV-2 Oxford AstraZeneca vaccine takes advantage of this pathway by coming in at the DNA level. The scientists used a weakened virus as a vector. This virus was from the adenovirus family that typically infects primates and rarely infects humans. Using this kind of adenovirus as a vector, rather than an adenovirus that infects humans, significantly reduces the chances of the vaccine being ineffective, which could occur as a result of the recipient having prior immunity to that specific adenovirus. So the scientists remove the genetic material from the inside of this adenovirus, modify this genetic material, i.e. removing the genes that would allow the virus to replicate within our cells, but at the same time, they also removed a region from the SARS-CoV-2 genetic material, the region that codes for the spike protein. They then combined this modified adenoviral genetic material with the genetic material from the SARS-CoV-2 that encodes the spike protein only. This hybrid modified DNA is inserted back into the empty adenoviral virus. These modified weakened adenoviruses can now be used as a vaccine. Once this vaccine is administered, these viral particles can infect human cells, but are unable to replicate and cause any significant illnesses. Instead, what happens is that the modified genetic material is read by our cells. And as you recall from the beginning of this video, the spike protein DNA is converted into mRNA, and then, ultimately, the spike protein of SARS-CoV-2 is produced. The production of this spike protein allows our cells to present this to our immune cells, such as CD4 positive T cells, a specific type of T cell. These T cells can then release Th2 cytokines. Th2 cytokines facilitate the differentiation and activation of B cells. Activated B cells produce specific antigen-neutralizing antibodies. So now, upon viral particles entering our system that contain the spike protein, i.e. the SARS-CoV-2 virus, these antibodies can bind to the viral particles, neutralizing them, stopping them from binding to and infecting our cells, resulting in a fast response to the infection, reducing the chances of mortality and spread of the virus. So how effective is this vaccine? Well, let me talk you through some clinical trial results, published on the 8th of December. For those of you who want to dive into the data a little more, check out the description. I've referenced the research article there. Participants in this trial were randomly assigned the Oxford University AstraZeneca vaccine, which I will now refer to as the AZD vaccine, or a control vaccine, which is either saline solution or a meningococcal group conjugate vaccine. Overall, this study had 23,848 participants. Participants in the AZD vaccine group received 2 doses containing 5 × 10 to the power of 10 viral particles, i.e. the standard dose cohort. A subset of participants received a half dose as their first dose, i.e. low dose, and a standard dose as their second dose, and this was the low dose standard dose cohort. The equation they used to calculate vaccine efficacy, which shows how effective the vaccine could be given ideal circumstances and 100% vaccine uptake, is shown on the screen now, where RR is relative risk, defined as the relative risk of developing the disease for vaccinated people compared to unvaccinated people. The US FDA guidelines indicate that they would license a vaccine against a pandemic virus that showed at least 50% efficacy, and WHO have indicated a minimum efficacy of 50% in its target product profile. Participants who received 2 standard doses, vaccine efficacy was 62.1%, and in participants who received a low dose followed by a standard dose, efficacy was 90%. Overall, vaccine efficacy across both groups was 70.4%. Moving on to the side effects and safety issues. Overall, the vaccine had a good safety profile. out of the 23,848 participants, serious adverse events occurred in 168 participants, 79 of whom received AZD vaccine and 89 of whom received the MEN ACWY or saline control. 2 points I want to highlight regarding safety are as follows. A case of transverse myelitis was reported 14 days after AZD booster vaccination as being possibly related to vaccination, with the Independent Neurological Committee considering the most likely diagnosis to be of an idiopathic short segment spinal cord demyelination. Secondly, a potentially vaccine-related serious adverse event was reported 2 days after vaccination in South Africa in an individual who recorded fever higher than 40 degrees Celsius, but who recovered rapidly without an alternative diagnosis and was not admitted to hospital. The participant continued in the trial and received a second dose of the allocated vaccine without a similar reaction. So now for our conclusion. No previous trials have been published on the efficacy of a viral vectored coronavirus vaccine, and so this study provides the first peer-reviewed evidence that induction of immune responses against spike protein using viral vectors provides protection against the disease in humans, as has been seen in animal models. So overall, AZD vaccine has an acceptable safety profile and has been found to be efficacious against symptomatic COVID-19 in its interim analysis of ongoing clinical trials. So judging by this initial data, the scientists have evaluated this is a safe and efficacious vaccine against SARS-CoV-2, and so this can contribute to the control of the COVID-19 pandemic. And finally, please comment below on what you think about this vaccination program. I would love to hear your opinions.